We are here to deal with the topic Mendel's Law of Genetics, foundation of modern genetics. After the beginning of genetics, when we talk about the next step, what Mendel do is the laws of Mendel. Before moving forward to the laws of Mendel, let's discuss the terminologies which we will deal with in this genetics. First terminology is gene. So what actually is gene? Gene is a unit of heredity. When we talk about gene, gene doesn't mean the entire DNA sequence. It is a part of DNA. So it is a part of DNA where it codes for a specific protein. Now what is allele? Alleles are the different forms of a single type of gene. If we talk about the height of the person, then the height can be a higher height, that is a tall person, or a shorter heighted person, that is a dwarf. So dwarf and tall are the two alleles of the gene. Homozygous and heterozygous. Homozygous means both the alleles are of same type. Heterozygous means they are of different types. Genotype and phenotype. Genotype is the genetic makeup. When we talk about A, T, G, C, and ATGCs are the base sequences which code for a genes which is represented by TT, RR, Y, X. These are the symbols which we use for representing a genotype, and how that is observed, that is the observable trait, is known as the phenotypes. So these are few terminologies which we'll deal with when we start about genetics. First is law of dominance. First we read the statement. When two alleles of a gene pair are different, one allele can mask the expression of the other. The allele that is expressed is called dominant and the one that is masked is recessive. Now there is two important one. First you have to focus on the term mask the expression of. Mask means it is masking or it is suppressing the expression of the other. So there is one which is always expressed because it masks the expression of the other. That one is the dominant one and the other one is the recessive one. So when we talk about the dominant trait, it always masks the expression of the recessive trait. So again I am reading it. When two alleles of the gene pair are different, one allele can mask the expression. When we are talking about it is different, it means if one is capital T, another is small t. So the alleles are different. If there is T and T, capital T and capital T, then there is no laws of dominance because capital T and capital T are equivalent and both are dominant. So law of dominance is represented by the statement that when two alleles of a gene pairs are different, one allele can mask the expression of the other. The allele that is expressed is dominant and the one that is masked is recessive. Example, the trait is a plant height. The alleles are tall, capital T is for dominant and small t is for the dwarf or the recessive trait. Now, this is the genotype. Wherever there is capital T with capital T, both are equivalent and capital T is denoting for the tall height of the plant. So, the phenotype will be tall. In small t and small t is representing dwarfed, so the phenotype will be dwarfed. But the case is when there is capital T with small t. So capital T will mask the expression of this small t, so the plant will become tall. Okay. This is law of dominance. If we take another example, if one plant has a red flower and another is a white color flower, both are observed in the same type of plant, then if suppose we say that this red is a dominant and then white will be a recessive one. So what will happen is it is represented by capital R and capital R. It will be represented by small r. Always remember that the recessive traits are represented by small alphabets what we used for dominant traits. As we have used capital R in the dominant traits, so for white we have to use small r. We can't use small w and small w. We can't use this. This will be wrong. So the correct interpretation is small r and small r. So if there is capital R and small r, then these will segregate and form like this. So we will get capital R with small r. Now if we get capital R with small r, then capital R will mask the function or the properties of small r, and all the plant will have the property of capital R. The property of capital R was red. So the color of the flower will be red. If we take another example, if suppose it is in heterozygous condition, then the traits will be capital R small r and capital R small r. You will get capital R, capital R, capital R, small r and small r, small r. These are the three cases. Now here it is both are equivalent, so you will get the dominant trait. Here both are recessive, so you will get the recessive trait of white. But here it is a mixture of dominant and recessive. You will get the trait of the dominant one. So if you calculate the ratio is there will be two small r small capital R. So the ratio will be 3:1. This is the phenotypic ratio. Remember, phenotype is the expression. These red and white are the expression. So the phenotypic ratio is 3:1. If we talk about the genotypic ratio, then genotypes are capital R, capital R, capital R, small r, and small r, small r. The ratio will be 1:2:1. This is the genotypic ratio. Now we move to the law of segregation, that is the purity of gametes. This is the second law of Mendel. Now in this law he says each individual has two alleles of each trait, but only when allele is passed to the offsprings during gamete formation, the alleles separate during meiosis. Now if suppose there is capital T with small t, then according to this law, both will get segregated in the gametes. So there will be two gametes, one with capital T, another with small t. Now this is the Punnett square, which we can calculate for our own. So this is the Punnett square, which is denoting that there is one capital T, there is two capital T, small t, and there is one small t. This gives the phenotypic ratio of 3 tall to 1 dwarfed. So this is the law of segregation. If suppose the law of segregation does not exist, this is the square we have taken with capital T, small t. If the capital T, small t's are the parents and there is no law of segregation, then what will happen? If this will fuse, then there will be only one trait, that is capital T, small t. Means capital T, small t means tall. So there is no possibilities. We can say that there is no possibilities of getting a dwarfed plant. But actually it's not the case. We are getting this dwarfed plant. Small t is the dwarfed plant, and out of 4, 1 is the probability of getting a dwarf plant. 1 by 4 is the probability of getting a dwarf plant. But if this law of segregation doesn't exist, then there will be 4 by 4 probability of the tall plants. So there will be only tall plants, no dwarf plants would have existed. Moving forward to the third law, that is the law of independent assortment, it states that genes for different traits are inherited independently of each other, provided the genes are located on different chromosomes. If the genes are present on different chromosomes, then they will segregate. There is independent assortment. No one is connecting, or no one is controlling the segregation or assortment of the other traits. So here we have taken seed colors, that is yellow and green, and seed shape round and wrinkled. This is most of the common example which we ever take. So this is the ratios. Let's suppose capital R, this is the trait which we have taken. Capital Y, and this is small y, small r. And getting the ratio as 9 is to 3 is to 3 is to 1. F1, let's see. If we have capital Y, capital W crossing with small y and small w, then what will happen? In the F1 generation, we will get capital Y, capital W, and small w. And this will be selfing in the for the second generation. So when we will self it with capital Y, capital W, small w, what we get is the ratio of 9 is to 3 is to 3 is to 1. This we'll get with the help of the Punnett square. Okay, this will get with the help of Punnett square. Let's try to have this. Just a second. If we draw this Punnett square, then what we get is, first with their genes will segregate, or the gametes will segregate. With capital Y we will get capital W, capital Y with small w, small y with capital W, and small y with small w. These are the 4 types of gametes which we'll get in case of male and in case of females, because it is selfing. So when we segregate out, we will get a cross of 4 cross 4, getting total 16 varieties of this, and out of the 16 varieties, you will segregate it with the ratio of 9 is to 3 is to 3 is to 1. I'm giving the Punnett square cross in the link, so you can see it how the cross is made. We can draw here also capital Y, capital W. Then capital Y we get small w, small y with small w, or small y with capital W, and small y with small w. These are the types of crosses which we will get, and when we make this Punnett square, you will get the entire so results as 9 is to 3 is to 3 is to 1. Let's calculate it. We have capital Y with capital W, capital Y with small w, small y with capital W, and small y with small w. This is suppose of the male parents, and another we get for the female parents. As we are doing the selfing, so there won't be any. This is capital W and this will be small y and small w. So now we'll get first column, we will get capital Y and capital W. Here we will get capital Y, capital W, small w, capital Y, small y, capital W, capital Y, small y, capital W and small w. So I hope you all know how to make it. We are just writing what all the male gametes and the female gametes will fuse to form the combinations. After writing the combination, these are the combinations of the genotypes. So after writing the combinations of the genotypes, we will move forward with the phenotype. This will be capital Y with small y, capital W with small w, small y with capital W with capital W, small y with capital W, small w. Here we will get capital Y, small y, capital W with small w, so this one is the fully recessive one and this one is the fully dominant one. So if we talk about the phenotypes, then the ratio will be 9:3:1. But if we talk about the genotypes, in genotypes this is 1 and the last one is also 1. Okay. Now if we see these two are same. So the gene ratio will be 1:2, 2:2. This two are same. So this 2:2, 2:1. This is this is again 1. And now if we look at here, you will find these 4 are having same. So the genotypes will come to 4. And again we will find here this is 1, this is 2, this is 2 and this is 1. So the genotype of the F2 cross will come to 1:2, 2:2, 2:1, 4, 1:2, 2:2, 2:1. So this is the genotypic ratio. The genotypic ratio comes to 1:2, 2:2, 2:1, 4, 1:2, 2:1. Now when we talk about the phenotype, now how it is expressed? Phenotype will be yellow with round, that is one phenotype. Other one will be yellow with wrinkle. Next will be green with round. And the last one will be green with wrinkle. So there will be only 4 phenotypes. Now when we talk about the phenotypes, we have to calculate. When we start calculating, you will find capital Y and capital W. We will find here 1, 2, 3, 4, 5, 6, 7, 8, 9 places. So capital W, so it will comes to 9. Yellow with wrinkle will be capital W with small w, capital Y with small y. Now capital Y with small y, it comes here 1, 2 and 3. So it is coming as 3. When we talk about green and round, it is small y with small w. Again it will be coming as 1, 2 and 3. So it is 3 and 1 is green wrinkle. It is this one. So we have yellow is to round, yellow round with yellow wrinkled, green round with green wrinkle. So is 9 is to 3 is to 3 is to 1. This is the ratio what we get after the law of segregation. Now if we want to summarize it, we'll find that the law of dominance focuses on one trait. The dominant trait masks the recessive trait. Law of segregation focuses again on one trait where the alleles separate during gamete formation. And the law of independent assortment focus on two traits. Alleles of different genes assort independently. This is the most important traits, and in the competitive exams the questions basically come from the F2 crosses with a dihybrid ratios. These are the two traits. When we talk about two traits, it is a dihybrid. When we talk about one trait, then it is monohybrid. So this is the Mendel's law of segregation, Mendel's law of dominant as well as independent assortment. Hopes this explanation is sufficient to understand the Mendel's law. If you have any queries, you can give it in the comment sections. Thank you and happy studying.